Good evening, everyone. I'm Priyanshi, and today I'll be presenting about the research that I did as a Talaria scholar with the help of my mentor, Amelia Ryan. I wrote a literature review on the use of nanomedicine for early diagnosis of Parkinson's disease. Before I begin, it's important to understand the pathology of the disease and why is it relevant in today's world. Currently, it's the fastest growing neurodegenerative disease, and 10 million people worldwide are infected with Parkinson's disease and are living with Parkinson's disease and the kind of standard of living that Parkinson's gives us. But why does Parkinson happen in the first place? Parkinson's, in a very basic sense, occurs because of one trigger reaction, which can be caused by a lot of factors such as either inherited, environmental factors, etc. It occurs because of the death of dopamine neurons in the basal ganglia region of the brain called substantia nigra. As you can see in this image, it compares a healthy brain to a Parkinson's disease brain, where we can see that in the substantia nigra region of the brain, dopamine neurons constantly degenerate, leading to symptoms of Parkinson's to occur much later after the degeneration has started. And these symptoms are motor symptoms that include uh, tremors, bradykinesia, rigidity, etc., and non motor movements such as depressive disorder, olfactory deficits, rapid eye movements, sleep disorders, anxieties, etc. The present development of Parkinson is very limited in terms of both diagnosis and therapeutics. Currently, diagnosis happens much later after the degeneration has already started because the motor symptoms emerge much later after dopamine degeneration has started. And usually the diagnosis happens in form of screening. There's no lab tests that are actually conducted to make a conclusive diagnosis about Parkinson's. Doctors monitor tremor, exclude other forms of Parkinsonism, or see response to L-DOPA. It's often misdiagnosed, and it's often diagnosed much later after after the after the body has constantly been degenerated and have been in later stages of Parkinson. Therapeutics, currently there's only one efficient drug used for Parkinson's, which is known as L-DOPA. We can't indirectly inject dopamine into the brain of the patients because of the blood-brain barrier that exists between the patient's brain and our body. And that is why it's very hard to develop therapeutics for neurodegenerative disease because of the blood-brain barrier. However, L-DOPA, which is a substitute to dopamine, can cross the blood-brain barrier and thus is the only efficient drug that we have. However, there's also a major flaw here because L-DOPA does not stop the progression of the disease. It only helps with the high tremor, and in long term, it also causes things like hallucination and other diseases that can eventually lead to worse standard of living. We can see this in this figure particularly, right? Because in this figure, we see how in the clinical state diagnosis happens at much later state when the onset of Parkinson's disease has already onset of dopamine degeneration has become exponentially less, which makes it very hard both to treat and both to treat Parkinson's, both to diagnose Parkinson's, and also look at the trajectory of the disease itself for further innovation in the disease. And hence, early diagnosis and efficient therapeutics are very necessary for this disease. And that was the objective of my research. Largely, as I worked with my mentor Amelia Ryan, I did a literature review on nanosensors, which I'll be talking about later, for early diagnosis of the disease and how particularly can be used in order to create more better interventions in therapeutics as well. So, so before I begin with nanosensors, it's important to understand what biosensors are because that they form the basis of what nanosensors are. Look at it this way, right? Biosensors are basically uh, tools or electronic tools that we can use to detect particular molecules in our body, which are bio uh, which are biomarkers for diseases. Uh, uh, Say for example, this bioreceptor made of enzymes, cells, app meter, DNA, nanoparticles, etc., sticks to annihilates, which are often biomarkers for particular diseases. For example, dopamine for Parkinson's, or a particular uric acid in urine for other forms of diseases. They stick to these annihilates, and they only stick to these annihilates. And that is the thing about biosensors. They have to be extremely specific. If they recognize or stick to all types of annihilates, it would not work. And hence the specificity of these bioreceptors allows them to detect these molecules in vivo or ex vivo, and allows them to transduce this signal, process the signal and transduce this with the help of light, heat, etc., and generate electronic signals which can be then processed to display what amount of that molecule do the patients have, and then the doctors can calculate what the stage of the potential disease is. Biosensors are currently used everywhere for different types of diseases, from cancer to a lot of ex vivo diseases, in order to detect the disease. For example, a very basic example. Uh, 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 sorry. So now we're going to begin with what nanosensors are. Nanosensors, in particular, are very interesting for our research because, as I mentioned earlier, biosensors have to be very specific. If the bioreceptors detect all molecules, it would not be a very efficient screening process, and they cannot be trusted, right? But nanosensors are particularly interesting because nanomaterials are substances having structures smaller than 100 nanometers with physiochemical properties capable of affecting biological processes. Nanosensors, a tool of particular interest in the healthcare field, are devices capable of carrying data and information about the behavior and characteristics of particles at nanoscale level to macroscopic level, making them perfect for in vivo detection. And that is why there's been a current research going on in this field for diagnosis in this particular for the diagnosis using this particular technology. And that is why I explored three biomarkers. In this presentation, I'm going to be talking about three biomarkers of Parkinson's disease and how nanomaterials have been developed to target these biomarkers in particular. First, I'd like to begin with electrochemical ammeters with gold nanoparticles for alpha sign detection. Alpha sign is a biomarker for Parkinson's disease. What we see here is a Lewy body. Lewy bodies are abnormal cytoplasmic deposits with neuronal cell body loss. In Parkinson's disease, Lewy bodies are mainly found at the predilection site of neuronal loss, i.e., the substantia nigra and locus coeruleus. This has led to the conclusion that Lewy bodies are somehow related to nerve cell loss. Dopaminergic degeneration and the presence of Lewy bodies are two aspects that represent the hallmark pathology of Parkinson's disease. Lewy bodies lead to the creation of this, which is alpha-synucleic, a particularly conclusive molecular biomarker for Parkinson's disease. Alpha-synuclein is a 14-kDa protein found in the synaptic terminals of neurons in the substantia nigra part of the brain. Misfolded and aggregated alpha sign is a major protein component of Lewy body and is thought to be a pathological hallmark for Parkinson's disease. And that is why gold nanometers in particular are used to detect these biomarkers in order for early diagnosis of the disease. How does that occur? Is that recently, electrochemical immunosensor based on gold nanoparticles, modified graphene, has helped in ultra-sensitive and specific monitoring of alpha-synucleic. The gold nanoparticle conjugated ammeter was able to specifically recognize and detect alpha-syn with a detection of 10 p.m. This is a huge development because still now we are not been able to detect alpha-synucleic in vivo with the help of nanoparticles or any form of biosensors. This method has been used and experimented in various papers with specific modification, as I explored in my paper with Yu et al., Carboga et al., Yin et al., etc. Some of them use the presence of alpha-syn in CSF to detect a potential method of early diagnosis before symptoms arrive. The second the second very prominent way of detection is single-walled carbon nanotube space sensors for dopamine detection. As I already talked about how dopamine is a huge biomarker for Parkinson's disease and how its degeneration in the particular part of the brain can be used by scientists to characterize Parkinson's disease diagnosis in a more efficient way. And thus, single-walled carbon nanotubes are used. They're beneficial because they're composed of sp2 hybridized carbon atoms and can be rationalized as rolled-up cylinders of graphene sheet. SWC and D are small molecules which allow for recognition of variety of annihilates with specificity. Semiconducting single-walled carbon nanotubes are fluorescent in near-infrared ray due to their electronic, as you can see in this image, due to their electronic band gap between the valence and conduction band. The NRI emission range of carbon nanotubes coincides with the tissue transparency window, hence giving them an edge for in vivo diagnosis. And thus, carbon nanotubes have led to the development of various biosensitive detect and quantify biomarker for a number of disease and annihilates. So this particular figure is particularly interesting because it just shows us the breakthrough of using carbon nanotubes to detect dopamine in the most efficient way. This is Mann et al.'s paper which led to the development of carbon nanotubes, sensors for dopamine to a GFP bonding nanobody, and introduced the sensors to developing Drosophila embryos. The motor protein Kinase 5 was labeled with GFP, and aggregation of nanobody carbon nanotubes complexes on the protein was seen upon injection. In the study, carbon nanotube sensors also remained sensitive to dopamine, showing protein-targeted delivery of carbon nanotube sensor in vivo. Along with several other studies also looked at dopamine detection after this using carbon nanosensors, and hence it's a potential. This technology has a huge potential to develop and increase. The last bit that I like to talk about is alpha-syn oligomer and future therapeutics with nanomedicine. So alpha-syn oligomers are basically a toxic alpha-syn that are present in the substantia nigra part of the brain along with alpha-syn. And it is a more indicative biomarker for Parkinson's disease because it excludes any other form of Parkinsonism. It is something terribly unique to Parkinson's disease, and hence developing a particular carbon nanotube for this biomarker has been extremely tough because of the high specificity that is required. There have been current potential decreases, but there has not been any conclusive research about this. For example, Kachi et al. Designed a compact label-free sensor that can be used to detect and differentiate monomeric from a fibrillin alpha sync with high specificity. Since alpha molecules are known to be toxic species that can include Parkinson's, the detection of alpha-syn oligos in human plasma can also be useful. However, research in this field is still sort of not properly developed. In terms of therapeutics, we've also been using nanocarriers, which though have not been the focus on my research, I have explored a lot about it. Blood-brain barriers are a huge issue with Parkinson's disease, and nanocarriers, which are able to cross the blood-brain barrier and then release the drug, have been explored extensively for better therapeutics. Okay, the diagnostic and therapeutic intervention, as we've seen, for this disease is very limited and inefficient. Moreover, early diagnosis and effective therapeutic intervention can lead to way better prognosis amongst Parkinson's disease patients. Nanomedicine's unique ability to identify a wide diversity of annihilates make it optimal for early diagnosis. Several successful studies have been done in relation to this by identifying Parkinson's biomarkers such as dopamine and alpha-syn. The potential of nanocarriers as a drug delivery mechanism across the blood-brain barrier is also a major scope of study. Hence, we conclude that nanomedicine as a field provides a large potential for sensing diverse annihilates and creating an early diagnosis mechanism through detecting disease biomarkers. Thank you so much for listening to me. I'd like to give a huge shout out to my mentor, Amelia Ryan, for always supporting me, and also the Talaria team for giving such an amazing opportunity the research about what I'm passionate about. Thank you so much.